So after Mendel studied his data, he came to what we usually call two different laws or principles. The first one is sometimes called Mendel's first law, or the principle of segregation. It says that two alleles segregate or separate into different gametes during meiosis. And so that each gamete any gamete only has one copy of each gene. And then when two gametes fuse together during fertilization, it brings two alleles of each gene back together into the nucleus of that zygote. And now remember, the reason this is impressive is that he didn't know about meiosis. And so what he's describing here is what happens during meiosis 1 primarily, and then fertilization, bringing the alleles back together. So the cross really that I have already described to you can sometimes be called the monohybrid cross, although really not the whole cross is the monohybrid cross, really just this part of it here. So remember when he self-fertilized the F1 individuals and then took those seeds and planted them? That is really what we call the monohybrid cross. But if we look at Mendel's entire experimental design, in this picture they're representing the alleles with letters. And so this is typically what we do. So each pea plant, and they're representing the whole pea plant as just a yellow dot or a green dot. And that, in this case, is going to indicate whether or not the alleles that they're showing indicate whether the peas are yellow or green. So, so let's say you have a true breeding a pea plant that makes yellow peas. And you also separately have a true breeding plant that makes green peas. So in the pea generation, you have the yellow plant, I'm going to call it homozygous yellow, and then the green plant, homozygous green. And so those are your pea generation individuals, the true breeding ones. And so the yellow pea plant makes gametes that carry, the only allele they can carry is the yellow allele. The green pea plant makes gametes that carry the green allele. And so now in this case, they're indicating the yellow allele with a big Y and a green allele with a lowercase y. That indicates they already have reason to know that the yellow allele ends up being dominant to the green, which it does. So anyway, they're already kind of indicating that. Capital letter is the dominant allele, lowercase letter is the recessive allele. So the gametes from the yellow plant and the gametes from the green plant, when they fuse together, they can only make, it's not that they can only make one offspring, but all of the offspring that are made that way would all be the same genetically. They would all be big Y, little y. And since big Y, the yellow allele, is dominant, then they would have the yellow peas. And so then what they would do would be to self-fertilize this guy. So that's where you get that arrow. So the F1 plant that is heterozygous is self-fertilized. And when you self-fertilize the F1 hybrid, you'll notice that the F1 hybrid can make gametes that are, half of them will be big Y and half of them will be little y. So for the eggs, half would be big Y and half would be little y. And then since it's a self-fertilization, the sperm would also be half big Y, half little y. And so what we see down here is called a Punnett square, although Mendel didn't use a Punnett square. This came later. But what we're doing basically here is we're saying, okay, if a big Y egg and a big Y sperm fuse together, then you'd get an F2 plant that is big Y, big Y. If a little y egg and a big Y sperm fuse together, you would get an offspring that's big Y, little y. If a big Y egg and a little y sperm fuse together, you'd get an offspring that's big Y, little y. And if a little y egg and a little y sperm fuse together, you'd get a little y, little y. So this is just all the different things, because you have two kinds of eggs, not just two eggs, but two types of eggs in terms of which allele of Y they're carrying, and two types of sperm in the sense of which type of Y, the Y gene that they're carrying. And so you would expect to get, for every one offspring that's big Y, you would proportionally get two offspring that are big Y, little y, and you would to every one that's little y. And so that's the genotype ratio of the F2 generation. You would expect these genotypes in this ratio. So, you know, it's just a ratio. The phenotype ratio, since big Y and big Y, little y visibly look the same, they're both yellow, then your phenotype ratio would be 3 yellows for every 1 green. Because remember, phenotypes are always descriptive words. Okay? All right. So that would be what we usually say in the F2 generation. You would expect 3 that have whatever phenotype is from the dominant allele for every 1 that have the phenotype that is associated with the recessive allele. The 3-to-1 ratio. So here's Mendel's data. This is the actual data. You can look at his actual notebooks online. I think it's on mendelweb.net or mendelweb.com. But in any case, they have his actual notebooks. And of course it's in German. But you can see the numbers in there. And the this one I showed you earlier, 705 violet, that's purple flowers to 224 white flowers. And when you reduce that down to what's called the empirical ratio, the simplest ratio, it ends up being 3.15 violet for every 1 white. And you do kind of the same thing for each of these sets of data that he generated using the same experimental design. And what he noticed when he analyzed the data was that all of his ratios were within a reasonable error rate of being 3-to-1. So he saw that in the ratio. He saw that happening in his data. So if each individual has 2 alleles for any one gene, and if they segregate during meiosis, and if they then come back together during fertilization, then what that would predict for the monohybrid cross would be that out of those 3 individuals that were yellow, because remember, he couldn't see the genotypes, he can only see the phenotypes. So he saw 3 yellows for every 1 green. But it would predict that there would have to be 1 that's big Y for every 2 that were heterozygous big Y, little y. Out of the 3, it would have to break down this way, and if his thinking was correct. So what he did was he set up a test cross. I know the word test cross sounds like it could mean anything, but test cross is a specific kind of cross where you have an individual who has a phenotype that is the dominant allele phenotype, but you don't know what their exact genotype is. That's why on this one they put big Y and then a question mark, because we don't know what this second allele necessarily is. So he had a whole bunch of plants that were yellow, but he wanted to know how many of them were homozygous for the yellow allele and how many of them were heterozygous. So for each one of the yellows, for example, he would cross the yellow plant to a green plant. Now you know that the green plant has to be little y, or the green pea, the plant that makes green peas. So if you cross a plant that is big Y question mark with a green plant, and if you get all the offspring they look yellow, then that must mean that this question mark would have to be a big Y. If, though, you crossed a plant that had an unknown genotype to a plant that was green, and you got half of the offspring were yellow and half were green, it doesn't even have to be exactly, but some yellow and some green, then you would be pretty sure that this unknown allele in the parent was little y. So you're using the test cross to look at the offspring and decide what the genotype of that one parent, the full genotype of that parent, would most likely be. And that's how the test cross is used. Okay, so up until now we've only been dealing with one gene, like the gene for flower color. There might be, there are two alleles for the gene, but it's just one gene. But what about if you have two separate genes, like the gene for flower color and the gene for pea color? Those would be controlled by different genes on different chromosomes. Like maybe the gene for flower color is on chromosome 3 and the gene for pea color is on chromosome 7. And so Mendel did crosses that combined or looked at two genes at the same time in combination. So now we're going to start thinking about that. All right. And in order to think about two genes, how they would segregate in combination, you want to think about some statistics, some rules of probability. And two genes on different chromosomes are going to segregate and going to behave as independent events. In other words, the way that chromosome 3 lines up at the metaphase 1 plate in meiosis is completely independent of how the two copies of chromosome 7 are going to line up at the metaphase 1 plate in meiosis. All right? And I'll explain what I mean by that in a minute. But when you have two different events that are happening simultaneously, like the segregation of the gene for the segregation of the gene for flower color and the segregation of the gene for pea pod color, when those are happening at the same time, we say they're independent events occurring simultaneously. And when you have two independent events occurring simultaneously, the probability of two events that are independent occurring simultaneously is the product, meaning multiply, the product of their individual probabilities. So here's an example. If I asked you, what's the probability if you had a die, a little die, if you had a die, what's the probability that you would roll a 6 on the die? And if you had a coin, a penny, what's the probability you would flip a coin and get a tails? So I'm asking you, in one hand you're going to roll a die, in the other hand you're going to flip a coin, but you're going to do both at the same time. That's going to be a little bit tricky, but let's say you do that, okay? What's the probability that you would get a 6 on the die and a tails on the penny? Well, what you do is you just think of them separately. By itself, when you roll a die, what's the probability of getting a 6? Well, 1 out of 6, because there's 6 sides on the die. And when you flip a coin, what's the probability of getting a tails? Half. So the probability that if you did both at the same time that you would get a 6 and a tails, you use what's called the multiplication rule. Because whatever you get on the die doesn't influence what you get on the coin. So they're completely separate and independent events, but the probability of these two specific events, rolling a 6 and flipping a tails at the same time, is simply the product of their individual probabilities. So the answer is 1 over 12. So when Mendel dealt with two genes at the same time, he set up his experimental design exactly the same. He started off with plants that he double-checked to make sure they were true breeding, and those are the P generation plants. And so in this case, he has a plant that's true breeding for yellow peas, that's big Y, and also true breeding for round peas, big R. Then he had a plant that he had pre-tested that was true breeding for green peas that were wrinkled. So little y, little y, little r, little r. Now, even though these letters are next to each other, it doesn't mean they're on the same chromosome. Okay, so imagine the gene for color is on, what did I say, chromosome 3, and the gene for shape is on chromosome 7. Then when you cross the P generation plants, all the plants that you would get in the F1 would be hybrid or heterozygous for both traits. So big Y, little y, and big R, little r, all the plants would be like that. So you would have 100% yellow peas that are round. Big R stands for round, little r stands for wrinkled. And then remember, he self-fertilized those plants to get the offspring. Now, when you think about the plant that's heterozygous, I want you to think about it in terms of actual chromosomes. So here's a cell, or maybe a nucleus. You have 2 long chromosomes, maybe that's chromosome 3. One of them has a big Y, and one of them has a little y. You have 2 copies of chromosome 7, maybe that's a little chromosome. One of them carries a big R, and one of them carries a little r. So that picture represents this thing right here. So, and then this line with a little slash through it is the chromosome. So the only rule in the gametes, remember the gametes are going to be along the top of the Punnett square and down the side of the Punnett square. The only rule is every gamete has to get a full set of chromosomes. It has to get one of the long chromosomes, either one, and one of the short chromosomes. So if you notice, it's possible you could get a gamete that has a big Y and a big R, because that would be these chromosomes on the right, or excuse me, on the left. That's possible. We don't draw the chromosomes really. It's possible to get a gamete that has a little y with a little r, because that would be one of the big chromosomes and one of the small chromosomes. It's possible to get a gamete that has a big Y with a little r. Oh, that's kind of messy. And then it's also possible to get a little y and a big R. So you'll notice those are the 4 things they've written across the top and down the side of the Punnett square, because those are all the different egg types and all the different sperm types that could come from the F1 individual when they are self-fertilized. So then you bring those together inside the little boxes of the Punnett square, and you figure out the probability of the offspring. So what's inside the grid is all the different kinds of offspring that could come from the self-fertilization of the F1 individual. And the genotypes, there's all kinds of genotypes that are possible, but when you simplify it to phenotypes, out of the 16 possibilities in the grid, 9 out of 16 are going to have yellow peas and round peas, yellow round peas. 3 of the possibilities will give you green round peas. 3 of the possibilities will give you yellow wrinkled peas. You're supposed to see that this is bumpy. And 1 out of 16 would be green and wrinkled. So this is called the dihybrid cross. The dihybrid cross means that you're self-fertilizing a double heterozygous individual. So again, this part is the dihybrid cross, where you're self-fertilizing the hybrid. In this case, hybrid for 2 genes, or heterozygous for 2 genes, big Y and big R little r. But what this really is, what you're really doing is using the laws of probability. All right? So this is a trihybrid cross. I don't want to do a trihybrid. What you're really doing is using the laws of probability, because if you self-fertilize a big Y, little y, big R, little r to itself, then what you're really doing is you're really crossing a big Y, little y to another big Y, little y. And then at the same time, simultaneously, you're also doing a cross of big R, little r, cross to big R, little r. In other words, you can take apart, since the Y gene is on chromosome 3 and the R gene is on chromosome 7, and I made that up, I don't know for sure, but since they're on separate chromosomes, which they are, then you can take it apart and deal with it like it's two completely separate events. So instead of having one dihybrid cross, you have a monohybrid cross of big Y little y cross big Y little y, and you have another monohybrid cross of big R little r cross big R little r. So two monohybrid crosses that are occurring simultaneously. And that's much easier to work out, because if you have a monohybrid cross of the yellow and green alleles, then what you expect would be 3/4 of the offspring would be yellow and 1/4 would be green, because that's a 3-to-1 ratio is typical of any monohybrid cross. And for the R's, you would have 3/4 of the offspring would be round and 1/4 would be wrinkled. So then I would ask you, okay, what's the probability of getting an offspring that is yellow and round? Well, that is the probability of two independent events occurring simultaneously is the product of their separate probabilities. So I would multiply 3/4 times 3/4, and that would give me 9/16. So 9/16 would be yellow and round. 9/16. What would be the probability of getting an offspring that was yellow and wrinkled? 3/4 times 1/4. So that's 3/16 would be yellow and wrinkled. What's the probability of getting something green and round? Well, green is 1/4 and round is 3/4. So that would be 3/16 again for green and round. And then the probability of getting green and wrinkled would be 1/4 times 1/4. So 1/16. And so when you do that, you see where that 9:3:3:1 ratio comes from. Out of 16 possibilities, 9 would have the dominant trait for both of the different genes. 3 would have a dominant trait for one of the genes, but a recessive trait for the other. So round is dominant, but green is recessive. Another 3 out of 16 would be kind of the opposite. They would be dominant for the color, but recessive for the shape. And then 1 out of 16 would be recessive for both traits. So that's using the multiplication rule to figure these out. All right, so let's talk about the hybrid crosses before I get into this slide. If you have any cross that follows the general pattern of big A little a cross big A little a, that would be a hybrid cross, because both parents are hybrid, heterozygous. And it would be a monohybrid cross because there's only one gene involved, gene A. A dihybrid cross would follow a pattern. The letters don't have to be the same, but big A little a, big B little b, cross big A little a, big B little b. That would be a dihybrid cross. So that would follow the 9:3:3:1 ratio. All right, let me erase all that. And then a trihybrid would follow the pattern big A little a, big B little b, big C little c, and then cross big A little a, big B little b, big C little c. So this is the only pattern that is a trihybrid cross. If you have a different pattern, it doesn't have a name. Not every cross has a name. But Mendel's crosses, he did in a certain way, they have names. So this slide is showing you how to use, instead of drawing a Punnett square for a trihybrid cross, because it would be an 8 by 8 grid, you would have 8 gametes of each on each side of the grid. So it would be a 64 square grid. You don't want to do that. So you can use the product law to figure out the probabilities. So the way they do it is, if you're just looking at one of the genes, the offspring of any hybrid cross shows a 3-to-1 ratio. So if it's color, 3-to-1, 3 yellow for every 1 green, or I would do it in a fractional form, 3/4 yellow, 1/4 green. And then for round and wrinkled, 3/4 round for every 1 wrinkled. And then over here, 3/4 round for every 1 wrinkled. And then they're bringing in another gene now, tall and dwarf. Tall is dominant to dwarf in this case. In plants, by the way, it's different in humans. But you can see what I'm doing here. So if you wanted to know what's the probability of an offspring of a trihybrid cross being yellow, wrinkled, and tall, then all you're going to do is multiply the numbers, 3/4 times 1/4 times 3/4. You just follow the forked lines, you follow that path, and you can solve it. So it would be 9/64 would have the phenotype of yellow, wrinkled, and tall. What if I asked you the probability of green, wrinkled, and dwarf? Well, each of those has a 1/4 probability, so it would be 1/4 times 1/4 times 1/4, 1/64. Okay, the denominator on all these will be 64. So that's called the forked line method, but it really is just using the probability law, the multiplication rule that I told you earlier. The other kind of problem solving that you have to start learning how to do are solving pedigrees. And we're not going to get too deep into pedigrees, but a pedigree is a family tree, a genetic family tree that shows, typically it shows phenotypes. And so, but we're going to try to fill in the most likely genotypes, and some of it's already been done for you just for the sake of solving this one. But on any pedigree, a male is a square and a female is a circle. And a mating is a line directly between a circle and a square. All right. This is not a mating right here. Down here, this is not a mating because it's not a direct line. But this between 1 and 2 is a mating. This is a mating. This is a mating. Oops. And then this is a mating over here. All right. So let me erase some of that. All right, so we have some matings. And then the offspring of a mating will hang down from that line in some way. So these lines that go vertical, if there's just one child, then there's just one line, and then it's like the fruit on the vine. Like this one here, this square that's little a, that's the child of person number 1 and person number 2. And then that child mated with this person over here. And they actually had 3 children. So when you have this line hanging down and then it sort of branches out, then you can see the 3 children from that mating. And then 2 of those children had children. All right. So what you're going to be asked in these cases, well, it depends. They can ask you anything. Typically, this one is showing you orange is unaffected, meaning normal. So unaffected by some kind of unusual disorder. So unaffected means normal. Affected means they have some kind of unusual physical affliction of some kind or something observable. So this is typically considered I usually say it's always the focus on the unusual. All right, so maybe they have an extra, no, an extra finger would be a dominant. Let's see, how about maybe they have cystic fibrosis. All right, but typically you get the shaded. The affecteds are typically shaded. Usually the unaffecteds are left unshaded. In this case, they did put them as sort of a yellow color. All right, and they're telling you some of the genotypes. All right. In this case, all the ones that are unaffected have a dominant A allele, an uppercase A. So big A must be the normal allele. Because remember, dominant, it doesn't matter. As long as you have one of them, then that's normal. So little a must be the cystic fibrosis or the unusual. So you would have to have two of those to have the unusual or the affected phenotype. All right. So I'm asking you, what genotype would you assign to individuals 1, 2, and 3? All right. Now, they're telling you a little bit of something based on the color. So if they're blue, they're affected. And we said that to be affected, in this case, it seems that the affected allele is the lowercase, the little a. So the person 1 must also have that little a, little a. Little a, little a. Now, what about person 2? Person 2 is normal. They're unaffected, but they have a child who is affected. All right? Now, because they're unaffected, just from that information, you could think, okay, they could be big A, big A. But remember, big A is dominant, so they could also be big A, little a, and still look normal. The fact that they have a child that's little a, narrows that down for you because one allele, one A comes from mom and one A comes from dad. So the only way that this could work is if number 2 is heterozygous. So sometimes you're thinking about what are all the possibilities, and then is there some information in the pedigree that helps me eliminate one of those possibilities. And in a similar way for person number 3, they're normal, so they could be big A, or big A, little a. But we have some additional information because they have a child who is little a, that means that they must be heterozygous. So you're using some logic to figure out what the most likely genotype is of certain individuals on the pedigree.